neighboring populations. There's no mutations, mating is random, and there's no natural selection. And we've gone through and looked at how a violation of large population size leads to genetic drift, okay, which leads to, of course, change in allele frequencies. And also non-isolation with gene flow, how that can change allele frequencies. And although mutations really are not a major aspect here as far as changing allele frequencies, they are very important for creating new genetic material upon which natural selection or other forces can operate. And I want to really emphasize that these mutations, when they arise, they are totally random, totally random. They do not arise because of a need for them to arise. They're just random. However, should they be beneficial, then the environment will select for them. And when we get to natural selection, we'll talk more about that. Okay, all right, any questions before I start going? Okay, so we just looked at mutations. Let me get this started. No mutations, but mutations are important. Okay, now the fourth idea here is mating is random. Okay, so what we're saying here is no phenotype will favor mating with the same phenotype or whatever. And in most cases, however, most populations are relatively small, and therefore individuals do tend to have a certain degree of genetic closeness, a little bit of inbreeding. It's not necessarily the case that they have the same parents, but they may have had the same great-grandparents. And in that case, there's a higher probability that certain alleles can occur together. And so when close neighbors, close relatives happen to mate with each other, this is called inbreeding. And most small populations, as long as there's no migration out of that population, for example, we had a prairie dog town, okay? Prairie dogs are a type of rodent that lives in the ground. They don't occur here in California, but in the high plains where I grew up, they were pretty common, but they live in the ground in towns, okay? They have like a little bunch, they all live together. And some of these prairie dogs, of course, the towns are far apart, and they don't travel very much from one colony to another colony. And therefore, over time, the only mates that they have, and let's say that in this town, there's only 25 or 26 individuals. And therefore, over time, the genes are being shuffled among the breeding population that happens to be there. And so it's sort of impossible not to have a common ancestor maybe 3 or 4 generations back, and therefore this is a type of inbreeding. Now, what it does, inbreeding doesn't change the allele frequencies, okay? It doesn't change the allele frequencies, but of course, we can't always rule out with my illustration here of the prairie dog town. It's a small population, and therefore you can expect drift. But let's assume that there's no drift. Let's just assume that there's no drift. Still, what the inbreeding will do is it will lower the number of individuals that happen to be heterozygous, and increases homozygosity without affecting the allele frequencies. Okay, and let's just give an example. Some plants, if you remember Mendel's pea plants, they were self-fertilizing. And so you can't get any closer to inbreeding than that with breeding with yourself, okay? And that's extreme case. And so let's just look at it for example here. So we have a population of plants, because the inbreeding of that selfing usually doesn't happen with animals. But with plants, some plants, it's common. So we have a species here that is fully self-breeding. And let's say this population, first of all, we have here P is equal to 0.5 and Q is equal to 0.5. Under those conditions, then p squared plus 2pq plus q squared would give us 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessives. So if we're having self-fertilizing, then big A is mating with big A little a is mating with big A little a, and little a is mating with little a. So if we have some offspring, let's assume that each of these have the same number of offspring and the same number survive, but okay, and grow up. What is going to be the phenotype of the big A times big A? What will be the phenotype of the offspring?
Dominant.
Yeah, they'll be homozygous dominant. All right, and what about the little a?
Homozygous recessive.
Homozygous recessive, okay. And what about the big A, little a?
Will it be 50-50?
50-50.
Sorry, let me see. There's going to be 1 or 25% homozygous dominant, 50% homozygous recessive, and then 25% had homozygous recessive.
Exactly. So what we have here is that 25% of this offspring, okay, remember they started out with 0.5, all right, so 25% of that 0.5, so 0.25 times 0.5, that amount of offspring has to be added to big A. And therefore, if we kept this 0.25, okay, it's passed on to the next generation, but we're gaining big A from the heterozygotes. And therefore, the frequency of big A increases to 0.375. Likewise, the same percentage of 0.25 out of 0.5. So you have to take 0.25 times 0.5. That amount will become little a added to this. And therefore, the frequency is this going up. And our big A little heterozygotes, they decrease by half. Okay, and here we're assuming each group, each phenotype is producing the same number of offspring and the same number survive. Okay, if we take this then through yet another generation, then it's going to be the same story. Okay, we passed 0.375 down to the big A, the third generation. But of the big A offspring of the heterozygotes, we have 0.25 times 0.25 end up being big A. And so we have to add that frequency over here. And the same thing with the little a. And therefore, once again, our heterozygote frequency, it goes in half. And this keeps on going, okay? So this is an extreme example of inbreeding. Now, if you were to figure out so we have these allele frequencies, they still add up to 1. But you notice here that the frequencies of the homozygotes is increasing, and heterozygotes is decreasing. But the allele frequencies are the same, okay? If you have a calculator, this is my calculator, because to find out what P is, just a second here, let me get a pen. P would equal to this plus 1/2 of that. Okay, so that should be
0.5.
Yeah. So you'd take this plus 1/2 of 0.125, and what does it turn out to be?
0.5.
0.5. And likewise, Q is, if you did the same thing, is equal to 0.5. You notice here that the allele frequency has not changed, because up here, the allele frequency is also 0.5 and 0.5. So inbreeding doesn't change allele frequencies, but it does change genotypic frequencies. We have an increase in homozygosity and a decrease in heterozygosity. Still, that is a change in the genetic structure of a population. So this is how inbreeding is working. Now I'll give you some examples here. And this is based on a study that was done back in the, I think it was maybe in the 1970s or so. There was a small village that was studied in Africa. And all the individuals usually chose mates and married within the village, okay? And there was not a lot of movement outside of the village. Within this village, there was no type B blood, okay? So there was just two alleles, IA and little io. So the only possibilities here was that. With blood samples, they could tell an individual that was heterozygous, okay? Because an heterozygous individual, of course, would have type A blood. And so when they did this, looking at the alleles from, from the blood samples themselves, they determined that the frequency of big IA allele in that population, in that village, was 0.6, and for Q, it's 0.4. However, and so if you using those numbers, expecting these types of frequencies, then you would expect 36% of to have have type A blood that was due to the genotype big IA, homozygous, and 2pq 0.48 or 48% would be heterozygous, big IA, IO, and then little io, homozygous recessive 0.16. But when they actually did the blood typing of the individuals as to whether they are homozygous dominant, heterozygous, or homozygous recessive, this is what the genotypic frequencies turned out. And so we have here, you notice here that the frequencies for the homozygotes are higher, whereas for the heterozygotes, they're lower than what the Hardy-Weinberg formula would predict, okay? Now, in this case, of course, the allele frequencies are just a little bit off, because if I take 0.54 plus 0.1, it ends up with P being 0.64, but that's sort of close, okay? Sort of close. And of course, this is a small village, so you can't rule out genetic drift as being an influence, and we can't isolate that. But the best explanation, what is happening in this particular village, is that there is a degree of inbreeding, okay? A degree, not brother-sister, okay, but still cousins or grand cousins or something like that. And that is what has led to the genotypic frequencies being off from what would be predicted. Okay, all right. Another thing here with non-random mating is a phenomenon that is pretty common, is assortative mating, okay? So this could be positive assortative mating or negative assortative mating. And this is when the positive assortative mating is when individuals prefer mates of the same phenotype, okay? And in humans, we do have a little bit of that assortative mating going on. It isn't very often do you find a tall person married with a short person, okay? They usually try to be pretty much the same stature. So here, if the same phenotypes are preferring each other, then it's not random mating, okay? And it can lead to allele frequency changes and also genotypic frequency changes. Positive assortative mating, they're going for like phenotypes, but there's also negative assortative mating, where one individual of one phenotype would prefer to mate with a totally different phenotype. And so this is pretty common among animals. Okay, all right. So all those. Now, these first four Hardy-Weinberg conditions, they do not, they lead to microevolution, a change in allele frequencies and genotypic frequencies. However, they're non-adaptive. They're totally random, okay, in which direction the change happens to go. And when I say non-adaptive, it means that the changes do not necessarily lead to a better fit between the organism, the species, and their living conditions and their environment. Natural selection, however, is different. It leads to adaptation. because the environment is choosing or selecting the phenotype that best survives and can reproduce. And as long as this has a genetic basis, then those genes will increase in the population. Okay. Now I think I gave you all of this. This is my big definition of what natural selection is. It's differential survival in survival and differential success in survival or reproduction. Reproduction is a real big part here. First of all, an individual has to survive. That's important. They have to survive to reproductive age. But if they do not reproduce, it's a evolutionary dead end, because these favorable gene combinations that allow these individuals to survive, they have to be passed on to the next generation in order to be of any benefit. And so so it's not just only the ability to survive, it is the ability to reproduce. And even if we have two individuals that they can survive to reproductive age, but if one individual in this population leaves behind 10 offspring and another one leaves behind only 5, then the one that leaves behind 10 has a greater chance of passing their genes on into the evolutionary chain. All right, and therefore the ratios of the alleles will change over time. And so it is the population that evolves, not an individual, but still the individuals are carriers of these genes. All right, so this variation, phenotypic variation that the environment selects, it has to be genetic. Okay, if it's something acquired by exercise or something like that, it has absolutely no relationship to evolution. So, as I said, survival is only part of the game. Also has to lead to reproductive success. And so that's what's real important here. So what we have with natural selection, it's an interaction between the phenotype. And of course, the phenotype is due to the genotype. The genotype determines the phenotype, but still it's the phenotype that is interacting with conditions in the environment. And those that have a variant form that can better utilize resources in the environment and leads to reproductive success, they will tend to, in that particular phenotype and that underlying genes that encode it, will tend to increase over generations, whereas the other phenotypes will decrease. All right, so this leads to adaptation. Okay, the other 4 types of conditions, they are random. They do not lead to adaptation. They still can be important because, as I said before, if we have a favorable mutation that happens to arise in one, it always arises in just one individual. And that one individual, how can that allele increase in frequency when it shows up in just one individual? Well, if it's a small population, although it's totally due to chance, it can increase by simply genetic drift, after which natural selection can select for it. Okay, so, but we're still looking here, I'm trying to look at each one of these as isolated phenomena. All right, so, but this is the only condition, Hardy-Weinberg condition, that leads to adaptation. And so it's very, very important. Adaptation is a better fit between the organism and its environment. And what I mean by that is that that particular phenotype can utilize the limited resources better than other phenotypes that encourage its survival and reproduction. Okay, now emphasize, I already said this, individuals do not evolve. They're born with a set of genes, and those genes, they might mutate in the skin or something like that, but it doesn't really make any difference on future generations. It is the nature of the genes in the whole population. It's the population that evolves, and then the species as well. Okay, so emphasizing here, changing allele frequencies is microevolution. Okay, make sure everyone got that, because I'll move on to the next slide. Okay, I'm going to give you an example here. This is a very famous example. And what I have up on Canvas, I don't know if I have it published yet. I don't think I do, but I'll publish it today. I have a folder there that's additional readings. These are not required, but still they're scientific papers that I've gotten from various science journals that I have that pretty interesting to read. And at this stage, you would be able to understand them pretty well. And I have several of them on this particular story of the peppered moth. This is a species of moth that is in England, and it was widely studied. In fact, insect collections, we have insect collections that are in museums in Great Britain where this moth, we put them on pins and save them. They have them back before the Industrial Revolution, but this particular moth came in two different phenotypes. Now, this is not a single gene trait. Okay, it is polygenic, but still, that's a good example of natural selection. It has two different phenotypes, one that was dark and one was light. Prior to the Industrial Revolution, the light form was more prevalent. And during the Industrial Revolution, which began somewhere in the 1840s or so in England, the dark form became more prevalent, okay? And why is that? Okay, before 1850, it was rare to find the dark form, the dark phenotype. But afterwards, in some places, mostly in those places where there was a great deal of industrial pollution, because at the beginning of the Industrial Revolution, what was the energy that they used was coal. And so there was a lot of burning of coal and a lot of air pollution because of the coal smoke. Now, one of the things in industrialized areas happened with the coal smoke, the smog, is it killed on the barks of trees lichens. Okay, lichen, L-I-C-H-E-N. I'll go ahead and write that down. A lichen, as we will see a little bit later, is sort of a combined creature. It consists of a fungus that's living in association with a green alga or cyanobacterium. And the morphology of the fungus when they're living in a lichen form is quite different than when they're not, okay? But these things, it's sort of crusty little light colored structures that you find growing on the barks of trees or on rocks or things like this. Sometimes they can be very colorful, bright orange or yellow. Yellow and white are very common. So a lot of the tree barks in this wetter area of England were covered with lichens, but they're very, very sensitive to pollution. And therefore, with all this coal dust in the air, smoke, it killed off the lichens. Therefore, the bark turned. And furthermore, the settling of the smoke on the bark turned the bark of the trees darker. Right. So this is just a picture that I found. And this is of both moths are there. Okay, you can readily see the dark morph on this light colored bark. This is the light form right there, and it's not too easy to see. Now, if we have a lot of pollution, killing off the lichens and also coating the tree with the coal dust, we see now that the dark form is not very easy to see, whereas the light one is pretty prevalent. Okay, and this change probably had a lot to do with what was going on with that switch and increase in the allele frequencies or the phenotypic frequencies of the dark form. So scientists at the early part of the 20th century, Kettlewell, he did some experiments. And so what he did is in his laboratory, he reared and grew both the dark forms and the light forms in his lab. And then he marked them. Okay, so there's a way to take certain types of ink and put it on the wings of these moths in such a way that it doesn't hinder their ability to live, but nonetheless allows them to be identified. And so he would go out into some woodlands in Birmingham and also in Dorset. Dorset was an unpolluted area. It was far away from industry. Birmingham had a lot of industry and therefore very polluted at that time. And so he would release his marked moths, equal numbers, into the forest and come back several days later and catch moths. Now, of course, he's catching, and this is a common marked release procedure that entomologists frequently use to estimate the size of populations in woodlands or anywhere. So he went out, recaptured some. And of course, he's not going to recapture all of the released. He would also be capturing ones that he did not mark. And so he has both marked and unmarked, but nonetheless, looking at what proportion of his light and dark marked ones, okay, if he released them 50-50, okay, even numbers, he should be collecting them also in equal numbers, right? That's just the logic. But what he found is that in the Birmingham area, he was mostly catching the dark form. He had a higher proportion of his marked dark ones that he recovered compared to the light forms. Whereas in Dorset, it was the other way around. He was capturing a higher percentage of light forms than dark ones. And these were of his marked ones that he released in a 50-50 proportion. And so what caused the difference? So what he supposed is that these were camouflaged against their predators. These moths are attacked by birds. Birds are primarily, or certain species of birds, are primarily visual hunters. Okay, they see their prey. And what he reasoned is that in the polluted areas, the white moths were being picked off at a higher rate than the dark form, simply because the birds could see them better. Okay, they were less camouflage. Whereas in the Dorset area, it was the other way around. The light ones blended in with the tree trunks much better, and they were better camouflage. To verify this, what he did, let me see, do I have anything here? No. Okay, to verify this, what he did is, furthermore, is he took his moths out into both of these woodlands, and he tethered them to trees. Okay, and then he ran over here, jumped behind a bush, sat there all day long with a pair of binoculars, and really dedicated, and observed how birds would attack these moths. And so he pinned these moths to the trees, equal numbers, and he visually counted how many attacks on the light form versus dark form were done by birds. And what he saw is that in the Dorset or in the Birmingham area, where it was polluted, that the birds more frequently were picking off the white moths, the light form. And in the Dorset area, it was the other way around. Okay, and so what was causing the change in allele frequencies, or the change in phenotypic frequencies here in these two areas, is rest on camouflage. Okay, and so the dark form in the Birmingham area, it was more adapted, okay, to the dark form, the dark polluted areas. And so this is what adaptation happens to be. So this is a classic of how natural selection works, classic study. And later, some of the papers that I'll make available to you, there was other people later who challenged that this was really what was going on, and later other scientists. So I have several papers there, that one paper by a scientist that then answered the critic. And so it's a very interesting reading. Okay, so one of the things about natural selection, however, it doesn't create anything new. Okay, just natural selection by itself, it operates on existing variation, existing phenotypic variation. Okay, but and so one of the things, that's where the mutations are important. Mutations are creating new variants upon which natural selection can work on. So this is the only one of the Hardy-Weinberg principles that lead to adaptation. So this is a closer fit between the organism's phenotype and the environment, leading to greater survival and reproductive success. Okay, and as I said before, it's more than just survival. That's part of the game. And at natural selection, of course, they have to survive in order to reproduce. and it works on variation. And that variation has to be genetically endowed. Okay, make sure everyone has that. So this is just sort of a summary of natural selection. Everyone have this. Okay, if not, we'll come back. All right, so since natural selection happens to work on variation, really how much variation is there in real populations? Now with the peppered moth, of course, we just had two different moths, the light form, the dark form, and their phenotypic frequencies change depending on the changes in the environment. Incidentally, today with pollution controls in England now, the dark moth is very, very rare because pollution, we don't have that type of cold pollution there anymore. And the light forms have recovered because of this. Okay, but in a real population, how much variation is there? I'm going to introduce this term here called polymorphic. Morphos means morphology or form. And so when we say poly, it means more than one, okay? It's variable. We have, because in some cases, it might be monomorphic. There's just only one type of character, one type of form for a particular character. But here, we're looking for polymorphism. How much variation is there? Because that's what natural selection has to work on, is the variation. And this variation, we can look at it in many different ways. It could be anatomical variation, or now, since the 1960s, we have biochemical variation, which scientists prior to the 1960s could not observe, okay? Because we now can look at the variation in protein structure. We can look at the amino acid sequence of other types of proteins. We can look at the nucleotide sequence of genes themselves, and we can see this hidden biochemical variation. And so all of this, of course, is due to genetic variation. So we can measure it. And some of the measures that they use is a lot of times, especially the biochemical variation, is they will look at a number of gene loci, not of all, because like most mammals will have somewhere between 22,000 to 30,000 protein gene loci. And of course, we can't look at all of those. And so there's a sample of gene loci that are examined. Usually these are proteins. Yes, Savannah.
I'm just a little confused on what polymorphic means. It's more than one form of like genetic variation?
Yeah, well, if you're looking at anatomy, then it's having different anatomical differences. Okay, if we go back to the flowers, okay, since we had red flowers and white flowers, that's polymorphic. If it was a species that they always had red flowers, then that's monomorphic.
Okay.
Okay, and so if we go to biochemical variation, let's say that we examined in humans the nucleotide sequence for histone 1A. Okay, that's part of a nucleosome. All of us have exactly the same sequence, monomorphic. It's so that histone protein is so important in the folding of DNA that if it varies, apparently, if it varies just a little bit, it's fatal. Okay, and therefore it has been maintained. All of us have the same thing. But if we then look at the amino acid sequence of our alpha hemoglobin chain, we would find that there's some of us will have a certain amino acid at one position, whereas others have a different amino acid at that same position. That's polymorphic.
And if and so like if a flower had like, it could be red, pink, or white.
That's polymorphic.
Okay.
Okay, so this just means that whatever character you're looking at, that there's more than one form of it. Okay, if everyone is exactly the same, then that is not polymorphic.
And is it talking about just like, could it relate to like just specific traits in a living organism, or is it like for the entire population it has like different traits?
It can be both ways. Okay. Yeah, it can be both ways. Yeah. Okay. Aquazin.
Wait, so off of what Avani just said, so in an individual, can it have like what do you mean by it could go both ways with being polymorphic in an individual?
No, okay. I misstated that. I misunderstood the question. If she was asking about, can an individual be polymorphic for one character? No.
Okay.
Yeah. You have to be one or the other. Yeah. Okay.
So polymorphic is just among the variation.
Well, it depends. Okay. Yeah, it does depend. For example, if we're looking at allele or genotype, okay, or even let's stick with genotype. A heterozygote is polymorphic.
Okay,
But have different alleles.
But they'll still express one phenotype.
Yeah, exactly. Okay, so it depends on what level we're looking at what we're calling a character. If we're looking at the phenotype, then yes, it has to be just one phenotype there, an individual. But in the population, that phenotype, there may be more than one phenotype, and hence it's polymorphic. And but at the allele level, or at the gene level, something that's heterozygous biochemically, heterozygote would be polymorphic. And so that's possible. Yes, Avani, new question?
Yeah, I just want to rephrase this and make sure I'm understanding it. So a population can be polymorphic for a certain trait, and a population can also be polymorphic for like multiple traits.
Well, the population could be polymorphic for more than one. It could be 3, it could be 5, or it could be just two different variations in the character.
Like, so like if like a flower could be red and pink, and then like another trait could be like round leaves and pointed leaves, like both of those would be considered.
Okay. So if you're talking about different characters.
Yeah.
Okay. Yeah, you can have one character that is polymorphic and another character that is not.
Okay.
Yeah. So that's if you're looking at individual characters, yes, you have to visualize them independently of each other.
Thank you.
And you mentioned the histone 1A. That's not an example of polymorphism because everyone has the same thing in humans, right?
Yeah. Okay. Histone 1A. In fact, most of the histone molecules, they hardly differ between animals and other organisms. I mean, that protein is so highly conserved. So it says that, you know, if you deviate a little bit from allowing nucleosome to change, because these aren't very large proteins. I think they're, how many amino acids? I don't quite remember, but it's way less than 100 amino acids. They're relatively small proteins, and but apparently, and they're very rich in lysine and arginine, which carry a negative charge on their side chain. So it apparently that if you change one amino acid, it may lead to the nucleosome not being able to form correctly, and therefore you don't fold up the DNA. And if you don't fold up the DNA correctly, then that's usually fatal. Of course, those histones have also another property that's important other than just folding up the DNA. It's there are sites where acetylation and methylation takes place on certain ones of those, which can govern whether or not a gene is being expressed or not. And therefore, those are very important, and therefore they've been strongly selected for a particular amino acid sequence, and any deviation from it seems to have been eliminated. So we do have a few proteins like that. Others that, you know, they can be very variable. For example, I showed hemoglobin, beta hemoglobin, as an example of where we get evolutionary evidence of evolution. Our main amino acid sequence of this particular protein is hardly different at all from that of a gorilla. Okay, we differ by just one amino acid on average. But when we compare human beta hemoglobin to frog beta hemoglobin, it's quite a few more amino acids that are different They're quite different. However, that hemoglobin operates the same because certain amino acids can substitute for other amino acids, and the beta hemoglobin overall has the same shape in a frog as it does in human, or pretty close to the same shape. But with the histones, it seems like it's really tightly conserved. Okay, so yeah, and in fact, when we compare two humans together, we are our DNA sequences, we're mostly something like 99.9% identical. It's that 0.1% when you're talking about 6 billion bases. 0.1% of 6 billion bases is still a large number of differences, and that's where we differ from each other. Okay, Alcazim.
Yeah, I'm sorry, I'm not trying to go off on a tangent, but you mentioned the hemoglobin in frogs and humans. Although they have the same shape, would it be functional in a human if you have like a frog's beta hemoglobin? Because I know like other animals, like you could have a pig's heart in a human, like a transplant, stuff like that.
Yeah. With this particular thing, because of the amino acid sequence, it's more than just the shape. You know, your immune system would still see it as different, and it would attack it.
Okay, thank you.
Yeah, because it isn't the immune system doesn't only detect things on the foreign proteins on the basis of shape, but also on other properties such as the sequence of the amino acids, and so there very likely would be a immune response. Now, what's interesting is that which shows that all animals and organisms have a common ancestry, is that they've done this with yeast. Yeast is a single-celled fungus, okay, single-celled fungus, but bread yeast, Saccharomyces cerevisiae, is a model organism. It's a type of fungus that they've done a lot of research on. And there's certain genes, but they've taken the human version of that gene and replaced in the fungus, in the yeast, deleted the fungus version of the gene and putting in the human version of the gene, and it worked just fine. It doesn't work just fine all the time. I forgot what percentage, something like 50 or 60% of human genes they've been able to put into a fungus work just fine in the fungus. I don't remember the percentage. There's other times where substitution does not work, okay? But it's interesting, okay? And fungus, of course, is not an animal. It's further away from us, and so interesting. Okay, does that satisfy
Thank you.
Okay, so measuring polymorphism. Okay, two different approaches that researchers started doing. They would sample, okay, if they're gene loci based on proteins, the percent of loci that happen to have two or more alleles. You know, some of the gene loci are across all the samples within a species may be monomorphic. Okay, that's very common, but here we're trying to look at how much variation is there. And another way is, and these two approaches give almost the same results. If we have a sample here, we look at the average percent of loci that happen to be heterozygous, because if they're heterozygous, that tells us that at that gene locus, it's polymorphic. It has at least two different types of alleles, right? So these are two common ways. Now, back in the 1960s, with the invention of development of electrophoresis, scientists would take samples, tissue samples, and look at certain types of proteins, separating them by gel electrophoresis. And so I'm giving here an example here of an experiment like in Drosophila melanogaster fruit fly, looking for the enzyme alcohol dehydrogenase. Okay, alcohol dehydrogenase is part that converts ethanol into acetaldehyde, okay, which then can be converted into something else. Fruit flies, they have, this is an important enzyme for them because they're feeding on rotting fruit, and a lot of times in rotting fruit, there's a very high fermentation of alcohol there. But nonetheless, this is one enzyme that they would look at. And of course, what they do is from one individual, up here we have individual number 1, individual number 2, number 3, extracting the protein, putting it in this well, and then adding it to, and this is agarose, and putting electric current here. And of course, the protein, if it's charged, can then migrate. Okay, migrates according to its charge, according to size and shape. Now, an individual would only have 2 alleles for this gene, okay, for this protein that codes for this protein, alcohol dehydrogenase. And so in this one individual, what would you say about whether the individual is probably homozygous or heterozygous? There's one band.
Homozygous.
It's homozygous. This one here, it's obviously heterozygous because it has 2 different versions of the protein, okay? And number 3, since there's 2 bands, it must be heterozygous too. And number 4, heterozygous, 5, heterozygous, and so forth. Okay, but collectively in this population, what's the minimal number of alleles in the population for alcohol dehydrogenase?
Minimum is 1.
Okay, how many different alleles? Now, every individual, of course, because these are diploid, every individual would have 2 alleles. But according to this gel, okay, how many in the population, in the sample, what is the minimum number of alleles for alcohol dehydrogenase in this population?
Wait, so is it also 2?
Okay, well, would you say that this band and this band, are they the same? Do you think these are the same proteins?
Yeah, I do.
Yeah, because they all migrate to the same place. So they probably represent the protein product of 1 allele.
Wait, so is there 4 then?
Yeah, that would be 1. This would be a second, because it's obviously these aren't the same, exactly the same protein. They must have an amino acid sequence slightly different from each other because they're migrating in the same gel, in the same conditions, they're migrating to a different position. So there has to be a difference in them. And likewise here, this must be a 3rd allele. And this here must be a 4th allele. Okay, so minimally, there are in this population, this gel gives us evidence of the existence of at least 4 different forms of alcohol dehydrogenase. And since the protein is encoded from a particular allele, there's minimally in this population 4 different alleles, okay? Now, how do you visualize this protein? Because a lot of times when they put this in here, it isn't the case that they've isolated the alcohol dehydrogenase. There may be other proteins in here, but since this has, a lot of times what they'll do is to stain this, to see the band for only that particular protein, is to attach a dye to the substrate for the enzyme, because the substrate will bind only to the active site of that enzyme, and hence we can visualize the protein. Okay, now I said this is a minimal number. Why is that? I said,
could we have missed like other people in the population that might not have been in this test?
well, no, we take a large enough sample, okay, large enough sample. But is it possible that you could have 2 different isoforms? Okay, when I say isoforms, that means two different forms of the protein, of the same protein. That's an isoform. If we had, is it possible, is it always the case that we have a different amino acid sequence between 2 similar proteins, that they would necessarily migrate different from each other in a gel?
Yeah, it's possible. If
Okay.
there's like 1 amino acid.
They could, okay, let's say that we have 1 protein here that we substitute that in the protein, alanine was substituted by valine, okay? Both of these are nonpolar, and probably wouldn't change where those 2 different forms would migrate. So this individual that has just one band, we're counting it as being homozygous, but it could possibly be heterozygous in that the two isoforms, the two forms of the protein, happen to migrate to the same position, and therefore we can't see them. So the point is that sometimes when we have just, obviously when we have two bands, no question, that's a heterozygote. But this with one band, we're counting it as homozygous, but there's always the chance that it could have been heterozygous and the two bands co-migrated. When I say co-migrate, it means that they ended up in the same place, and therefore we only see one band. Okay, all right. So when these types of studies have been done with various insects, okay, these are isozymes. They're different forms of the same enzyme. In Drosophila, about 30% of their gene loci. Now, obviously, with these experiments, they didn't look at every single gene loci. Most of these were protein loci, and about 30% in Drosophila of what they tested was polymorphic. And so there's a lot of hidden variation. And other studies with other organisms come up with pretty close to the same number. About 30% of the gene loci within certain species happen to be polymorphic. So there's a lot of variation there. That's the whole point. Now, some of this variation can, in a species, can follow a geographical gradient. For example, here in California, a lot of times some of the insects that I studied as a graduate student, those in Northern California, same species, tended to be somewhat light colored, yellowish, whereas the same species that I looked at from Southern California tended to be very dark brown. Okay, so there's, and it seemed like there was a gradient here from north to south. So geographical variation. Particular species, even if it's the same species, they may have different variations in one geographical area than what they have in another. Now, if this geographical variation happens to follow some sort of gradient, okay, a geographical gradient of some sort, then this is referred to as a cline. Okay, and I'll give an example here of a cline. In fact, it looks like I'm going to jump to the next slide. Does the slide have more to it? Yeah, I'll come back to it. Okay, so this was actually a study that was done here in California from researchers at Stanford University, and that looked at yarrow, this type of plant that grows around here. So going over the Sierra Nevada, okay, so this is on the other side of the Central Valley. You go up the Sierra Nevada, because this plant is growing at various elevations along the Sierra Nevada, and hit the top peak, and then you go down into Nevada toward the Great Basin, okay, like around Reno. And so these researchers, they collected at various points, at various elevations, yarrow plants, measured their heights, and they noticed here that there's a clinal variation. The higher elevation, the shorter the plants. And then the plant stature increased as you go down to a lower elevation. Now, this is curious. Now, could this be genetic? It could be environmental.
Could that have to do with like oxygen and stuff like that?
What's that?
Like the availability of like resources in the atmosphere, like oxygen or
yeah, or you know, water content and stuff like that. So variation that goes along a geographical gradient of some sort, in order for that variation to be significant for evolution, we need to ask whether is it due to genes or is it due to something else? So how would you go about determining whether this phenomenon that we're seeing here with the yarrow plant, whether or not it is due to genes? Is it genetically determined, or is it due to some other environmental factor?
Can we see the differences in like the percent of the loci at each elevation?
Let's say we don't have that type of sophistication.
We plant one at a different location and then see how they grow out, like the same seed.
Okay, say that again.
Like we plant the same seed at different location, like the same breed.
Okay, you're getting close. You're getting close. That would do it too, but that's a little bit complicated.
Can we just take one of all of them and then grow them in the same elevation?
Exactly. Okay, and this is what they did. So they gathered seeds from these plants and these various locations, took them back to Stanford University in a greenhouse, and grew them all in under the same conditions, same water, same sunlight, etc. Okay, and what it turned out is those that were gathered at a low elevation still turned out tall. Those that were gathered at a high elevation still turned out short. Okay, so that says that the variation is genetically endowed. Now, what would be the selection pressure that would lead to short plants? What do you think would lead to the evolution selecting for a plant that grew not so tall?
Maybe like something that eats it, like a certain animal at each elevation.
Yeah, that possible.
Like I was thinking of mountain goats or something.
What advantage would being short
Like
have?
wind?
With what?
Wind?
Could be.
When it's like shorter, like the plant, is it more conservative and consumes or uses less like energy, so it's going to stay short to survive at the higher rate?
Okay, how long is the growing season at high elevations? Because you're close.
Sorry, what'd you say?
How long do you think the growing season is at high elevations?
Is it? Are you asking Josue or can anyone answer?
The higher the elevation, the shorter the season, because winter sets in at high elevations very early. So the growing season is very short. If you went up to the Sierra Nevadas right now, you would find it covered with snow. And that snow is not going to show any bare ground for, into maybe June or so. And so things can start growing somewhere around June, but then it starts snowing somewhere maybe in October, or at least it gets too cold to grow around October. So the growing season is short. So it takes time for a tall plant to reach this height. Okay, it has to put in a lot of cells, and only when it reaches a certain height does it flower and produce seeds. If you move this plant here up into the high elevation, it would freeze before it would ever flower. It would not reproduce. And therefore, this selects for plants that grow very short and reach sexual maturity and produce flowers at a very early rate. Okay, and so that seems to be the explanation here. And this isn't just only true for yarrow. It's for a lot of plants that are growing at high elevation. They're stunted. They're growing very short because they have to reach sexual maturity. If they grew taller, they would freeze. Their winter would set in on them before they ever had a chance to reproduce. And if they don't reproduce, then they're not contributing any genes to the next generation. And so these are the forces that seem to have been working here. Now there's other clines that I can talk about. This is pretty common if we look at mammals that have big ranges going all the way from Texas up to the Yukon territories in Canada, is that many mammals, and this is true of hares, bears, and others, that the body size increases with latitude. Okay, what do you think? And so this is not just in one animal. Coyotes are like that. Hares are like that. Hare is a type of sort of a rabbit, but it's not a true rabbit. They both belong to something called lagomorphs, but they still have big ears. And bears, okay. Why do you think we have this type of body size variation as you increase in latitude?
Is it because it's colder, so they have to like?
Exactly. It's a conservation of body heat. Not only do we find that the size increases because because you produce heat as a function of volume, and you lose heat as a function of the surface area. And as we went over last semester, if you don't change the shape of the object, if you increase its dimensions, body volume increases as a cube, of linear measure, and the surface area increases as a square. Therefore, body volume is increasing quicker. And there's more to this too, because what we have, like with coyotes, those down in Texas have long ears, long legs. Okay, so there's also a change in shape. Whereas those in more colder regions to the north, they have shorter legs and shorter ears, because that is altering body surface area. Okay, Aquazim.
Can we say that race is also due to decline?
Say what?
Can we also say that like race is due to decline in the amount of melanin in your skin due to like?
Yeah. There is a relationship there. Yeah, there is a relationship there. Now, darker skin is it's more protective, it looks like, against ultraviolet radiation, but that isn't the whole story. It also seems to the melanin, higher amount of melanin in the skin seems to have something to do with the processing of folic acid. Okay, it's better for that. Moving to, as humans moved into from Africa, because all humans, Homo sapiens originated in Africa and moved out of there about 80 to 60,000 years ago. And we'll cover that a little bit later. But in northern climates, especially in Europe at 40,000 years ago, it was far more cloudy than it is today. Okay, very much cloudy. And that vitamin D production, it happens in your skin. Okay, the production, so there's a relationship between UV light and interacting with your skin to produce vitamin D, which is really a hormone. We'll find out a little bit later on. It's not really a vitamin, but nonetheless is very important for the production of bones and calcium metabolism in your body. So as humans moved into northern areas, they got less sunlight. And so that favored, and furthermore, it's cold, wear furs, okay, covering up a lot of the skin so that there's not too much skin that's exposed to the ultraviolet radiation. And therefore, light melanin skin, however, is much, much better at producing vitamin D. And so that seems to be the selective advantage here. Now, the first, as I mentioned yesterday, the first humans that ended up in Europe, they in 40,000 years ago, they had darker skin. And the type of lighter skin that we find in European populations is due to a mutation in a melanocortin receptor that's in melanocytes in your skin. And in fact, I think I have a science article on that, and that I have in Canvas, and you can read about that. Now, that explains European lighter skin. Those in northern Siberia, okay, they also have lighter skin than Asians that are living further south. Okay, so there is a relationship there. However, the genetic basis for the lighter skin in East Asians is not the same mutations that occurred for Europeans. And so it's convergent, okay, to a certain extent. Okay, so you're right. A lot of that pigmentation, the amount, and it's just the amount of melanin that's deposited in the skin that it had that type of thing. Now, that type of selective pressure now no longer exists, because we have moved, and we've removed ourselves quite a bit from a lot of environmental pressures that our ancestors in the past had to suffer through or deal with. Because if we cover up all of our skin completely, we can take supplements, okay? And if you understand what I'm saying. Is that?
Yes, thank you. That makes sense.
Yeah, okay. All right, so body size. Now, I want to point out, although I've been talking about natural selection on discrete characters, really what natural selection works on is the whole phenotype. And sometimes that leads to a compromise, because what would be bad about or disadvantage of having a large body size in Arctic regions? There's an advantage, and there would be selection for larger body size for withstanding the cold environment. But there's a problem with that too.
It takes more energy.
Okay, more food. Okay, yes, it takes more energy, and you have to get that energy from food. And the resources for food in the Arctic regions is not as good as it is in more temperate areas. And therefore, that would select, that environmental factor, the lack of resources to grow on, food and energy sources, would push in the opposite direction. Okay, it would select for smaller size. So what do we end up being? It's compromise. And this is exactly what evolution, natural selection leads to a compromise, not the best of all worlds, but to a compromise. Okay, because it operates not just on one character, the phenotype, it works on all. Okay, all right, so that's good. All right, so there's a lot of variation out there. Okay, about 30% biochemical among most organisms. But if it's the case that if we have two different phenotypes, whether it's a biochemical phenotype or an anatomical phenotype, and one of them is favored by natural selection and the other one is not, wouldn't that eventually lead to everything being monomorphic? Okay, that's a question. First of all, before I get to answering that, let's just ask, how is this variation even generated? Where does it come from? Okay, obviously, sexual reproduction, shuffling the genes, that combines alleles from two different parents, which can create different variations, phenotypic. Now, of course, and this is probably, and I mentioned this last semester, this is probably why sexual reproduction evolved to begin with, because there's some single-celled organisms such as the Chlamydomonas, you meet that in the lab. It's an algae, a single-celled alga that lives in ponds, freshwater ponds and pools of water. They usually reproduce by mitosis all the time until the environment becomes stressful. Okay, temperatures are starting to drop, or other things like this. And that's when they undergo sexual reproduction, because the sexual reproduction shuffles the alleles and creates phenotypic variation. And that we already learned about that last semester. And of course, connected with sexual reproduction is recombination, because we're really the phenotype, a lot of our genes, or a lot of our aspects of our phenotype are not determined by a single gene. They may be polygenic. And therefore, we shuffle the alleles from different genes by crossing over. Okay, recombination, we talked about that. It happens during meiosis. And furthermore, unequal crossing over, that can lead to gene duplications. And gene duplications seem to, in evolution, has happened quite a bit. Once we get duplicated genes, a lot of this unequal crossing over is due to the plentiful transposon sequences that we find in our chromosomes. We now have two copies of the gene, and sometimes one of them will mutate and become a pseudogene, but in other cases it mutates and the two genes diverge in their properties. And this is how genomes evolved. Furthermore, the ultimate source, however, of new alleles is mutations. And mutations, they happen. They're random. And these mutations, there's a certain rate at which they occur. And that seems to have been adjusted by evolution as well. Organisms have evolved systems to repair some of these mutations, but some of them get by. If the mutation rate were higher, in fact, I read an article not too long ago, if the mutation rate were higher, then the number of genes, essential genes that an organism would have, would drop to something like 8,000. And so the genome would have to shrink because the mutation rate would be too high. If the mutation rate were slower, then we could have a higher number of genes, but then evolution would slow down. So mutation is important for creating new genes, new alleles, However, I want to emphasize that mutations do not mutate in the direction in which the organism needs to adapt. They're not adaptive. What happens is that the mutations create a new allele. That new allele creates a new phenotype, and the new phenotype is tested by the environment, regardless. And it's the environment, the ability to survive better in that environment with that change is what selects for that allele, okay? Natural selection, okay, so it's rare, random. Okay, just to review a little bit about these mutations. Some of them are point mutations, they're harmless. Others can make a big change in the phenotype. And really it is the environment that will favor the new allele. Furthermore, environments change. We might have a point mutation that is neutral, okay, it's not harmless, or it's harmless, doesn't hurt the phenotype. But if we change the environment, it may now become favorable, okay? So these things can change. And even a mutation that is deleterious, that is not favorable, can be maintained by the environment if it really depends on the environment. An example of this is sickle cell anemia. Okay, so this is in the beta hemoglobin. We talked about it last semester. There's a 1 amino acid substitution at position 6 of the beta hemoglobin, such that under oxygen stress, the beta hemoglobin and the alpha hemoglobin, instead of forming a tetramer, they will stretch out into a long chain and therefore stretch the cell, and the cell cannot carry oxygen. These are red blood cells. So that's pretty bad, okay? However, in Africa, and I'll come back to this, where malaria happens to be, okay, areas where there's high amounts of malaria, this sickle cell allele is actually selected for, because individuals who are heterozygous for sickle cell, they survive malaria much better. Whereas homozygous normal, they have a higher fatality rate when it comes to malaria. And so the heterozygote is being selected for. And we find that the sickle cell allele is there at a higher percentage allele frequency than we would find in other places. Another example here is flies that became resistant to DDT. Now DDT can be, by some flies, they're that P450 oxidases that are in the ER, endoplasmic reticulum. These types of enzymes, certain forms of them can detoxify. But when we first started using DDT, most flies could not withstand it, and it killed them off. However, the flies that happen to have certain forms of these oxidases, they grew more slowly than other flies, and therefore they had a slower reproduction rate, and therefore were not favored. But once we put in the DDT, they were favored. So my whole point being here is that something that at first happens to be not favorable can become favorable if you change the environment. And so this change the environment led to now flies that can detox DDT. Okay. All right, so gene duplications, this is another place, I already talked about that, by which variation can occur. The globin genes in vertebrates, I've only mostly been emphasizing beta globin and alpha globin, but there's other types of globin genes that have other functions. There's one type of globin gene that is makes up part of the hemoglobin in the fetus, and then once the individual is born, then that gene is shut down and replaced by beta globin. All right, so gene families, that creates new genetic information. And a lot of times it isn't just only the protein encoding genes that are important, but also the regulatory sequences. And so last semester we talked about promoters and enhancers and so forth. So these regulatory regions, which don't code for protein, sometimes mutations in them can change the rate and timing of when a particular gene happens to be expressed. And that may be very important. Okay, an example here, I don't expect you to remember this word because that's sort of a, but this here is the example of a, not a gene, but a regulatory sequence that in embryos is normally expressed, that protein is expressed in the formation of your hands. Okay, and chimpanzees, the amino acid sequence of this protein, which guides the formation of your fingers, for example, is the same in chimpanzees and humans. Okay, the amino acid sequence of the protein is exactly the same. But there's been a mutation in the regulatory sequence that controls that gene, such that in humans, when they're developing, it's expressed earlier and in more cells all the way up the arms and for a longer period of time than what it is in primates. In primates, it's shut down very early. And it's believed that this may have something to do with the dexterity of our hands. Okay, same protein, but because protein is being made earlier, lasting longer, it has led to a different type of morphology in our hands that makes us more dexterous. Okay, and so this here protein, I forgot what it is. No, this is a sequence, once again, a regulatory sequence, and I should have looked it up because I've totally forgot what it is, so we won't worry about that. But it's another example of, in fact, this regulatory sequence is completely lost, and it's led to, between humans and primates, led to a different, outcome. I'll have to look that up and I'll correct it and I'll tell you about it on Monday. Okay, so this is the question I asked earlier. Right now, I'm saying here, where does all this variation come from? How is this created? Well, point mutations, sexual reproduction, gene duplications and mutations, they all are creating new variations. But if natural selection is selecting for one particular type of phenotype against the other, you would expect over time that the unfavored phenotype should disappear. So how come we have so much variation? That's where I'd like to go. How is this variation maintained? Because it's obviously maintained there. And so there's several different mechanisms by which, even in the light of natural selection, we still can keep these. Okay, one of it is the fact that we're diploid. Okay, so some of these deleterious genes, alleles, happen to be recessive. So they're only selected against in the homozygote recessive individuals, because that's when their phenotype is expressed. But the allele is still, and here we're talking about a case where it's complete dominance, okay? So in a heterozygote, they would have the dominant phenotype, although they have the recessive allele, and therefore it will not be selected against. Now, under these conditions, if we have a large enough population, of course, that recessive allele will still be at very, very low frequencies, but it's hard to eliminate it completely. All of us humans, in our genome, we have about 30 recessive alleles, somewhere around there, that if it occurred in the homozygous recessive state, it's a lethal allele. It would kill us. And about 30 of them. But they're down in very, very low allele frequencies. This is why probably societies in the past put taboos on brother-sister mating, okay? Because in that case, it increases the probability of forming a homozygote individual with that would be lethal, okay? We all have about 30 of those. Another way in which genes, alleles, can be maintained in the population is by something called balanced selection. And I gave one example already, is that of sickle cell anemia. So this is one form of balancing selection. Now, in the case of the sickle cell anemia, there's 3 phenotypes. There's individuals who are wild type, that they have normal hemoglobin. Those that have sickled phenotype, they're heterozygous, and those who have homozygous recessives. The homozygous recessives, where they have 2 alleles for the sickle cell condition, usually die early. They have a higher fatality rate. In malaria infected areas, like here. Okay, I have a map here. It's showing the areas where there's malaria, and this is the frequency of the sickle cell allele. Now it's calling the sickle cell gene. I don't like that. It's really the sickle cell allele. But you see the frequency especially in this area here around the Congo, it can get up to 13%, very high, okay? Whereas in other places where there is no malaria, then it is quite low. So here, this is heterozygote advantage, because the ability of a heterozygote to survive malaria and reproduce is much higher than the wild type or the homozygous recessives. And so this is what we mean by heterozygote advantage. Yes, Alcazar.
For the heterozygous of sickle cell anemia, isn't it still like something else is still expressed? I forgot if it wasn't the like full sickle cell anemia, but there's still something that's being expressed, right? With the heterozygous.
That's a heterozygote. Yeah.
But like it's like but isn't it also still affecting them?
Oh yeah. Yeah, under oxygen stress, they do have some of the red blood cells do sickle like this. Now, this is why they have a higher immunity to malaria. Doesn't mean that they don't get malaria, because what happens with the malaria parasite is that they get inside of the cells and they're feeding on it. And then at certain periods, they will burst out of the cell and find new red blood cells and invade them. If they happen to invade these sickled cells, it's a dead end, because those sickle cells, they're constantly being removed, and therefore the parasite cannot survive in them. If they get into a normal red blood cell, then yes, they can proliferate. But if we have, let's say, 50% sickled cells, 50% normal cells, then there's a less probability of the malaria parasite being able to survive there, compared to if you had a higher rate of, if you had a higher rate of sickle cells, then the individual is just so anemic, if it's a homozygous recessive, so anemic that usually they have a lot of organ failure and usually do not survive to adulthood.
Thank you.
Does that get it?
Yeah, that explains a lot. Makes sense.
Yeah, okay. Okay, and then let's do just one more. Now, it's another balancing selection. Actually, to tell these stories, I'm going to stop right here, because it takes too much time. So we've seen the theme happening right here is we have, just to summarize it before we quit, in the light of natural selection, we would, if we have one phenotype that's being selected for, another phenotype that's being selected against, you would not expect that we would have so much variation. Okay, variation should be reduced. We have about 30% variation in most populations, and so how is this variation being maintained? And so I'm getting into mechanisms whereby unfavorable alleles can still be maintained in the population. One of them is diploidy. In this case, a recessive allele that is unfavorable can hide out in the heterozygote. Okay, that requires it to be complete dominance. Another one is balancing selection, heterozygote advantage, where we have incomplete dominance, 3 different phenotypes, 3 different genotypes, but selection is on the heterozygote. Selection could be against the both of the homozygotes. And therefore, since the heterozygote has both of the alleles, the dominant and the recessive, they're both maintained and keeps our population heterozygous. And we'll go into others later. Okay, any questions? Any slides you need to see? Aquazim.
Sorry for that, Lloyd.
Okay, yeah, your hand was up. Okay, any questions? Any slide you want to see? We'll go ahead and quit here.
Can I see slide 44, please?
Which one?
44.
44. Okay, so everyone have a good weekend.